Hey friends, welcome back to my channel. So in last lecture we discussed membrane protein. Now let's continue with lipid linked proteins. So proteins are covalently attached to lipids. They help in anchoring proteins to the lipid bilayer or membrane. Types of lipid anchors. First one is isoprenoid linked proteins. So over here the lipid 15 carbon or 20 carbon chain is attached to C terminal cysteine residue via thioester bond. Next, fatty acyl linked proteins. So it is of two type. First one, meristylation. Over here meristic acid 14 carbon is linked to N terminal glycine via amide bond. The second type, palmitolation. Palmitic acid 16 carbon is attached to cysteine via thioester bond. Fourth one, GPI anchored protein. So GPI is linked to outer face of plasma membrane via amide linkage. So all four type of lipid anchors, their attachment and location. So here is the diagram. This glycine is linked to the lipid bilayer through amide linkage. Cysteine is linked through thioester linkage, and this is the GPI linkage. Okay, so next is hydropathy plot. This plot is to identify the spanning of protein, or we can say transmembrane spanning. So a hydropathic plot represents the hydrophobic and hydrophilic region of protein along its sequence. So in a protein sequence, how many residues are hydrophobic and how many residues are hydrophilic? If hydrophobic residues are more, then we can say that protein is alpha helix and it is spanning the membrane. So each amino acid has a hydrophobic index. Positive value indicate hydrophobicity and negative value of amino acid indicates hydrophilicity. So this is the hydrophobic index. So x-axis represent residue number and y-axis hydropathy index. So value greater than 0, these are hydrophobic region or hydrophobic amino acids residues, and the value less than 0, they are hydrophilic amino acid residues. If high positive peak greater than 0, that means hydrophobic region, and deep negative value or less than 0, they are hydrophilic region. So a stretch of around 20 to 25 hydrophobic amino acid is likely transmembrane alpha helix. This is very important. So membrane thickness is around 30 angstrom. Alpha helix rise is around 1.5 angstrom per residue. Required residue around 20 to 25 amino acids. Hence hydrophobic stretch of around 20 residues span the membrane. So how many residues are required to span this membrane is around 20 residues. Important feature, transmembrane segments are mostly non-polar, hydrophobic and often form alpha helix. So these 2 points are very important. Next, hydrophilic region found outside the membrane and interact with the aqueous environment. So peaks above threshold indicate membrane spanning domain and multiple peaks indicate multiple pass membrane proteins. That's all for today. See you in the next lecture.